have a better N50 value, that means the reads are assembled in a better quality. Okay. Now, suppose you have 50 million reads that are generated from a transcriptome data, and we know that there are roughly 25,000 to 50,000 genes in an organism. Okay. So your aim is to convert those 50 million reads into say 50,000 or slightly above than 50,000 contigs. Okay. So that you can get exact size of the genes. If you are getting very less number of reads, that means you have some problem in alignment, or you have given very stringent parameters of overlapping. And if you are getting very high number of reads after assembly, that means you are using very low stringent conditions, and your overlapping is uh, very low number. What is that? We will discuss in the coming slides. So N50 measure is basically tells you how good is your assembly. Okay, it is half of the sequences that are above than N50 value, and half of that they are low. Okay, last time we discussed that if you have 50,000 sequences and N50 value is 1 KB, that means that 25,000 of sequences they are more than 1 KB, and 25,000 they are less than 1 KB. Then here you also apply your biological sense that we have the genes that are approximately 1 to 1.5 KB in size. Okay, average length is this one. So we have some genes that are larger in size, CDS, not the exon and introns. Okay, so they go up to 2-3 KB, but they are very few genes, and we have very few genes that are less than 500 base pairs. Okay, so if your N50 value is coming like around 800, 900, that means it's okay. If your N50 value is coming around 500 or 1.5 or more than 1.5, that means there is something happening, that you have to change the parameters of assembly, or you have generated the sequences that are not able to make larger contigs, like you have not made very good parent libraries, or your library size is larger, and you are not able to sequence the gaps in between the sequences. That's how you will get N50 values, and then you try to reduce the further number of contigs by aligning them with each other. We have programs to do that, but nowadays I will explain in the coming slides that nowadays people, they don't try to reduce the number of contigs, because we have many splice variants. Otherwise we have problem in losing out the splice variants in that. Okay. So similarly, we have for genome assembly, you have NG50. So for genomes, larger is the NG50, larger is better one, because we have very large size of genomes, but it should not exceed the total size of genome data you have. Okay. So if you have a genome size known, okay, then it's always better. So people try to find out the genome size before sequencing the genomes, so that they come to an idea that how many reads they need to be generated, how much coverage they will require in the sequencing. Okay. Like for Arabidopsis, we have 100 MB, like I told yesterday, last time also, that for wheat we have more than 1 KB. Okay. Now, I think we discussed this last time also, that Illumina, produces very high yield, cheap, reliable. Read length is, nowadays it's long enough. They are producing the system that can go up to 500 base pairs. Okay, and the industry standards, they are very high, and most of the people, they use Illumina for this reason only. Okay, and the problems are that they have a GC problem. Then the regions, they are high in GC content, then the read quality lows. Okay, dipping of quality at the end of the reads, that is a problem with all the sequencing platforms, not with Illumina. Sanger also do the same. Okay, long running time for high seq. Now they have next-seq and other sophisticated platforms, and short insert size. Still, when we are dealing with the larger genomes, we may require longer reads. Then we have Pacific Biosciences that produce up to 5 KB data on an average. Okay, so you can sequence 5 kb of your DNA in a stretch. Okay, and the problem with this is that it has high error rate on the longer fragments, and it is still an expensive technology. But the problem is to skip all the library preparation, purification steps in the Pacific Biosciences. Then in nanopore, I told you last time that this is a very small instrument, but the error rate is very, very high. So that's why most people, they don't prefer it, although this is a very cheaper machine. Okay. Then when you assemble the data, suppose this is your black line is your reference sequence. If you have a reference genome, and these are your reads that are sequenced from both the ends, pairwise sequencing, then you should have a chance that every fragment should be sequenced independently. Okay, because there is no influence of this fragment on this or this on this. So you have every chance that each fragment should be sequenced independently, and you want all the fragments should be sequenced. For example, when you do like this kind of PCR, bridge PCR, so what happens in Illumina that you have your adapters and your fragments that are aligned on the Illumina, and you have bridge PCR. Okay, so on a plate you have several clusters of DNA. Then there might be problem that due to PCR errors, one cluster is very big in number and another cluster is very small in number. Okay, then the cluster that is very big in number, that have high probability of more reads will be sequenced compared to the cluster with low reads. But we don't assume that. We want that each k-mer is the fragment. Each k-mer has equal chance of being sequenced. Okay. Then when you generate this data, okay, then you want to create the overlapping of the reads. Okay, now you have small reads like this. Suppose this is one read, and you filter out adapters from both the ends, and then you check the quality of the data, that what is the Phred score of this. So each base score is calculated, and if you feel that the ends, like I said, that at the end the quality is not very good. So if you feel that at this end or at this end there is some low quality sequences, then you can trim it. You remove 10 bases from this side, 10 bases from this side, or more than that depending upon the quality, and then you assemble these contigs with each other. So how do you do that? So suppose this is your another contig. Okay, so you try to overlap this. Okay, so the sequence that have overlapping sequence, they will come this and align, and then another read will go like this, another will go like this, another will go like this, and you have generation of the larger contigs. Now there is a problem. Okay, so suppose you have given you want, like as a human being, you want that I should create a longer contigs. Okay, so what you will do, you will overlap the sequence like this. Minimum amount of bases should overlap. So it will generate large contigs, but that may be possibility that some non portion, ठीक है, कोई ऐसा केमर आपके साथ जुड़ जाएगा जिसका उससे कोई लेने देने नहीं, because you have given very low overlapping value, 4-5 बेस बाई चांस एक दूसरे के साथ मिल गए, and then they start making contigs. And another possibility is that you want to give very high stringent condition, that you don't want any unknown contig to be generated or unknown read to enter in that. So you keep the alignment parameters like this. So there is more overlapping sequences. So this will be very high threshold value. Okay, so what will happen in this case? You will lose the data. Okay, suppose you want to have highly accurate sequences and you have kept overlapping very high, then you will have smaller contigs, but they will be highly accurate one. Okay, then you will have very large number of contigs. Okay, so in this case you will have longer contigs but less in number. In this case you will have smaller contigs but very high in number. So you have to find out ideal situation, not like this, not like this, okay, where your assembly will be ideal so that you have an appropriate size of contig. Also the data is significant. Okay, so that we try on the computer by giving different overlapping parameters. Okay, so for this you have to do some basic information. Okay, we have like programs, Trinity assemblers there, and many other programs are there that are freely available nowadays. So they uh, make graphs for checking the quality of the sequence, like I said, pred score checking, read checking, you know, adapter trimming, all these are there. So they make De Bruijn graphs for Lebanon data and Sting graphs for the PacBio data. So these tell us how to set the parameters for an assembler, like this or this. Okay, the type of error that assembler makes. Okay, sometimes, like I said, we are humans, we want to generate longer contigs for our safety, but that may be some kind of erroneous alignment. Then why variants are lost? Okay, suppose like I said that after assembly we try to reassemble the data, okay, on each other, and then want to reduce the number of contigs. Okay, suppose I have generated again 50 million reads, and when I assembled first time it comes out to be 100,000 reads. Still I feel that this is very high number. I am focusing on 25,000 to 50,000 reads. Okay, and then you reassemble that data. Okay, you want to reduce the redundancy in that context, and you simply align those 100,000 contigs with each other and try to align the contigs and reduce the number. But what happened in this case, when you try to reduce the redundancy, the variants are lost. Why variants are lost? Suppose you have created a splice, or your organism is making a splice variants. That is everywhere nowadays. We know that splicing is present in all the organisms, and in Arabidopsis we have a splice variant data. Okay, and in Arabidopsis at one gene make at least 3 splice variants. Okay, so that means if you have 25,000 genes in the Arabidopsis, including splice variant, it will come out to be 75,000. So when you do reassembly, okay, so if you have splice variant, it will consider that this is some kind of assembly error, and it will throw out the splice variant and just keep the longer one. So you are losing the splice variants. Then some repetitions, they are collapsed because if you have some kind of repetitive elements in your genes, okay, it may be microsatellites. Then again you have problem that if you have some different microsatellites on 2 different alleles, then they may be lost during the assembly. Okay, then heterozygous regions may appear twice. Okay, because you have 2 different alleles. Okay, one may be from one source, another from another, and they will appear twice. So you have this kind of problem. Okay, this is again what I explained you in the overlapping. That overlapping fragment we call as a Kmer size. So you have to choose the Kmer size depending upon your data. So approximately when we align the data, we start from different Kmer size. Like we first try to use 20 base pair overlapping and see how many contigs it is making. Then we use 25, 30, 35, 40, 45, up to 50. So 30 to 50 is the ideal parameters where you do overlapping, and then see how much contigs they are making. And sometimes we get overlapping 35, 40, and that generate very good number of contigs. So Kmer is basically the overlapping size, okay, that are used to assemble the data generated by the Illumina or any other read program. Okay. So the smaller K-val value allows locating more overlapping sequences with higher sensitivity and increasing number of ambiguous repeats, like lower specificity. So if, like I told, if you have smaller value, it will generate more overlapping sequences. If you have lower comer size, it will generate low overlapping sequences. Then deep brain graphs, they are fundamentals for Illumina data. Like this is again the assembly. So it measure all the parameters automatically. So you don't need have to enter the sequence. So it will do overlapping of the alignments on the basis of Kamer size at its own. You just simply tell the range that from this range to this range you want to overlap, and it will do the same. Okay, so it will get one sequence, another sequence, so all the sequences they are aligned together, and then you have overlapping sequences. Okay, so if you have fragments like this, so this will be overlapped one by one with each other. Okay, so progressive alignment overlapping is there. Then if you have more redundant data, okay, so then you can increase the Kamer size or reduce the Kamer size depending upon the type of data you have generated. Okay, then FastQC, another tool that is most commonly used when you are not using Illumina Trinity software. So it is used to do quality assessment of the NGS data, that how accurate your NGS sequencing has been done. Okay, so it generates the QC, quality control plots. Okay, and but it doesn't have the ability to customize or interact with the plot. So it will be your input. It will only like compute the data and give you output, and then you have to see. For example, this. Okay. Now this is the quality analysis of the sequence. So this is a read of 50 nucleotides that has been sequenced by the Illumina, and this signifies the Phred score. Okay, so in the last lecture we understand that anything Phred score, anything above 20 is considered to be good when you are doing sequencing. So you can see in the beginning you have very good Phred score in the terms of yellow lines. So you have 28, between 25, 30, and moment it goes to the end, your Phred score plot widens up. That means there is some kind of problem in sequencing. So by looking at this graph only, you come to know that how much trimming you require. So here you can see up to say 30 you have quality that is more than 24, and then you are going down. So you cut from here, you cut these fragments. So you have a tools available where you say that trim 15 bases from all the reads from 3 pi end. Okay, at 20 bases or 25 bases. So roughly when you trim 20 bases, you are left with only 35 sequences. Okay, so 35 sequences in millions, and you have to assemble to make the larger data. Okay. Then you can interplay between FASTA format. Okay, so when you do practicals, you come to know that we have a FASTA format in bioinformatics, and you have FastQ format in NGS. Some programs they take FASTA format for analysis. Have you done BLAST or not? Okay, there you might have been told that this is a FASTA format. Okay, then you have FastQ format. FastQ format is the format generated by the high-end sequences. Like I explained last time, it starts with the @ sign. And some of our tools like BLAST, it doesn't take the FASTA format. It doesn't take the FastQ format. It takes the FASTA format. But we have tools available with you that you can convert your contigs into FASTA format or FastQ format. It's very easy. You can change that, and many commands they are written there. Okay. Then you want to do assembly. After checking the quality of the sequences, do you want to do assembly of the sequences. Now we have seen the parameters that are ideal for sequencing. Okay. Then you have trimmed the adapters, and then you have trimmed the sequences also that are having low Phred score. Okay. and then you want to assemble the data. So if again you are using a Trinity software, it automatically do everything. It has everything. It can check the quality. It can do alignment. Okay, but if you are using some other tools or machines like Solid or 454, then you have to use other tools. Like we have APSYS, GAP4, Genius, Mira, Nebular, S6, Obnovo, Wilbert. These are various tools that are available for alignment of short reads to make the larger contigs. Okay. So you have SOAP de novo. Again, they do the same kind of assembly like we discussed. We have Velvet, S-Sake, okay, Novo line. So they do de novo assembly or reference based assembly. So what is reference based assembly and de novo assembly? Reference based assembly is that you have a reference genome and you want to assemble your reads on that. So that's very easy. So suppose you have this is your reference genome and you have short reads generated, they will automatically go to the location where they are matching. Okay, so and everything is very clear. But what is de novo? That you throw your data and they will align automatically without any reference genome. So that's a little bit tough task. So that will align on the basis of the similarity at the ends. Okay. So that will be challenging, might be the overlapping is just by chance if you have not given very stringent parameters, and you might have problem in analyzing the results. Okay. So once you made the contigs, then you have to do the analysis. For example, I have mentioned here de novo analysis, or sorry, data analysis for mRNA data. So if you have sequenced the transcriptome, okay. So if you have sequenced the transcriptome and you want to analyze that, so if you have a reference genome, you can directly map onto that. Okay. So that will tell you the locations of the genes on the genome, that on which chromosome or on which region of the genome your genes they are located. Okay. And then you do analysis with the BLAST or BLAT files. Okay. BLAT file is BLAT is the program that is used if you have a reference genome available. Okay, it will align your sequences against the reference genome. BLAST is like we have done normal BLAST analysis. So normally first thing we do is do the assembly and do the BLAST analysis or BLAT analysis, so that you immediately come to know that how many known genes they are present in your transcriptome. Okay, how many known genes they are present in your transcriptome. So what happens, like when we sequence the transcriptome, so 50% of the genes they match with the database. Okay, they match with the database, and you directly come to know that okay, the 50% of the transcripts or contigs that we have generated, they have a similar sequence in database, and you immediately assign them the functions. But you have now novel transcripts also that are not matching with the database. Okay, if you are using BLASTX, normally we use BLASTX, not BLASTN, because BLASTN you give you huge amount of similarity without any information. Okay, only BLASTX tell you where the genes are. So if you have a novel transcripts that are not matching with the database, then you have to align them. Okay, because annotating the 50% genes that are not matching the database is a tough task. Okay, so you have to assign different functions. So then what do? You go to different databases, like you go to Go database or KEGG database or reference sequence database, or in the protein database search for domains they have, and then you try to assign them the functions, and then you also try to find out the splice variant, that how many splice variants per gene it has. So what do you do in this case? So you take a reference RNA. Suppose you have one gene that has matched to the database on the basis of similarity, and you come to know that, okay, this gene is doing that function. Now you want to find out the splice variant of that. Then how will you do that? You take it as a reference sequence and then align all your reads into it. So the reads that will align to that data, that sequence will be the splice variant. So you will see some gaps are in there and some are matching. So you immediately find out. So what do you do in this case? You make your sequence at a database sequence and then compare other reads with them. So actually, you don't want to work with all 35 or 50,000 genes. You choose some genes. Otherwise, make aligning one by one to 50,000 genes is very tough task. Okay, we have softwares you can do with that also. Okay, and then you have to again check the variants manually. That is a huge task. Okay. So depending upon the tool you use, you can see pretty big difference between even the same genome. Okay. Also, like I told last time, that I always say my students that don't trust a single software. Try different softwares. Try different alignment methods, assembly methods, and tools, because two programs, they give totally different type of data. So we have these short read aligners that are most commonly used. So this Bauti, this is one of the most commonly used program by many. जितने भी आप जहाँ भी इससे भी sequencing कराते हैं, they try to use, if you are asking them to sequence and assemble, they use Bauti. Then you have this SOAP or SOAP de novo, then MAC, a Novo line. So we have used many of them. All of them, they work very good. So SSEC, Velvet, Bauti, they are good softwares. Okay. Then speed and sensitivity. Okay. So that is again a parameter that you should consider in mind, that the assembly should not take very large time. Normally it takes 3-4 days to 1 week. When you put these reads into assembler, it takes minimum 2 days to 1 week, depending upon the amount of data you have generated and amount of overlapping sequence you have put into them. But it should not take endless number of time. Okay. So Bauti and SOAP, they are the fastest one. That's why I said that Bauti and SOAP, they are most commonly used, because they are they are fast, and they should be accurate also. Okay. So you have to check the accuracy also, not only speed. Sometimes in the case of speed, you miss some good alignments. So the program should be fast as well as accurate. Okay, so many of them, they are fast, also accurate. Then after you do this, you do alignment. Okay, suppose I want to analyze my genome. Okay, I have a reference genome available. I will sequence my genome, and जो पहले human genome sequence है, उसके साथ मैं alignment करूँगा, and then I will see how much differences I have with the genome, है ना, जिस person का हुआ था. Okay, this is comparative genomics, and mostly people, they are doing now this. So you have a one standard genome available, and you have different species or varieties of that, and you compare with the standard reference genome and try to find out the differences. Okay. So you find out the each reads that matches to, but this is not the purpose. We want to find out the dissimilarity between two organisms. That if I want to compare my sequence with any of your sequence, the things I will be interested in, how much we are dissimilar from each other, where we are differing, and that's why we are looking different from each other. Okay. So due to high sequence similarity between number of same species, most reads should map to the reference genome. Like if I will compare two human genomes, almost 99.9% of these reads, they will match. Only 0.1, or if you have sequencing error, or if you have low quality data, that will be remained. Okay. So how to map billions of reads into genomes? So you have these aligners, they align to make larger contigs, and you have chromosomes, and it will match to that location. So if you have a back library, or if you have done hierarchical sequencing, then you have reads, you have back clone, and you have the chromosomes. You can do like this manner. So if you have done shotgun sequencing, you have the reads, and then you have to search for a reference genome for correct assembly. Otherwise, you have to generate again back clones to find out the exact location of the sequences on the chromosomes. Okay. So this Trinity, it has 3 programs. Like I said, Trinity is most commonly used because it is free, and mostly people, they do Illumina sequencing. So if you are doing Illumina sequencing, you can use Trinity, very simple to use. So it has 3 programs. One is for quality checking, one is for assembly and splice variant analysis, inchworm, chrysalis, and butterfly. So this inchworm and chrysalis, they do uh, Dubrin graph construction, and butterfly, it tries to find out the isoforms or splice variants. Okay. So this we have discussed that you have to choose the right k-mer and construct these contigs carelessly. If you have used this, because you are not knowing what is happening inside. So it is just overlapping the sequences and then aligning it. Okay. Contig must correspond to most abundant isoform, because like I said, if you have a cluster on the Illumina which that is highly means PCR से उसके बहुत ज़्यादा है ना वो बन गए हुए हैं fragments. So then you have very high amount of sequences of that in the sample. तो उसका मतलब हम डायरेक्टली एज्यूम करते हैं कि अगर एक जीन ज़्यादा एक्सप्रेस कर रहा है तो उसका एक्सप्रेशन बहुत ज़्यादा है ना सेल में, अगर कम कर रहा है तो उसका एक्सप्रेशन कम आ रहा है. तो दैट वे डायरेक्टली correspond to number of reads. So if one gene is giving more reads in the data, that means the gene is highly expressing, and if it is giving less reads in the data, that means it is lowly expressing. But अगर कहीं PCR एरर हो गया ड्यूरिंग ब्रिज फॉर्मेशन, तो हमारा सारा जो है जो एनालिसिस है वो गड़बड़ हो जाता है. दैट्स व्हाई वी रीकन्फर्म दैट व्हाट हैज बीन प्रोड्यूस्ड बाय इल्यूमिना, है ना, उसको हम रीकन्फर्म करते हैं बाय रियल टाइम PCR that whatever it has done is correct or not. Okay, because whatever it has generated is not of guarantee, because there may be some kind of errors. So it tries to make clusters of the reads by overlapping them with each other, and these are the short reads that keep on aligning, and they try to make one long stretch of the contigs depending upon the parameters you have set. Then butterfly try to find the isoforms. Okay, in between, suppose there is exon skipping or some base changing, or 2 different leaves from 2 different parents are there, they try to find out that. Okay, and then you do assembly by using different k-mer sizes, that is overlapping parameters. You can check it and finally you do that. So now if you have assembled the reads, so these are, again to summarize, these are the steps that you should follow in the transcriptome analysis. Okay, suppose you have sequenced a transcriptome, the first thing that you do is quality assessment and filtering. Okay, so you should first check the Phred score of the data, then you select the k-mer size and you select the base trimming. Then for assembly and clustering, you have many programs like SOAP de novo, Velvet, Eland, that align the or assemble the data on the basis of k-mer size and Debruin graphs. And if you still want, like if you still feel that you have very high number of contigs and that are not expected, then you try to reduce the redundancy of the contigs by using the programs like TGICL and CAP3. They were used 4 or 5 years back. People always use this TGICL or CAP3 to reduce the number of contigs. But nowadays no one uses that, because if you will use that, it will remove the isoforms from your data, or it will remove the splice variants from your data. So nowadays, mostly people they don't use it. Then after you did बंद हो गया बैटरी लो हुई. अच्छा मेरे से नहीं ले लेना. So you do blast normally for transcriptome data. If you have a good quality workstation with you, it will take 15 days to 1 month to complete that analysis. Okay, so it will continuously run, and then you will get the files. And then after you annotate, you have, like I said, 50% read that will match to your database, and 50% that will not match to your database. So for rest of these, you should go to Go, CAG or EC database and try to find out the domains, motifs that tell you that it might be the function of that transcript that you have. Then another thing that we do in this is, like I said, we can quantify the expression, that if the one gene is expressing more, it means it will have more reads in the data, and if one is expressing low, it will have less number of reads in the data. So you quantify them by reads per million fragments. So first we do this and make the heat graphs, and then again, like I said, this may be PCR artifacts, then you reconfirm them by doing the real-time PCR. Then this is again some of the parameters that we normally do just to ensure that everything is in line, and just to put the bulk in your data, we measure the GC content. Okay, mostly the GC content is around 50% in an organism, so it should remain between around that. But some of the organisms like Mycobacterium, it has more than 60% of GC content. If these are there, then there might be the data that is, because Mycobacterium is a small organism, you can get the data, but sometimes you get more error in that. Then for like marker analysis, for example, for SSR, we use MISA software that can identify the SSR, simple sequence repeats, or microsatellites. These are the markers in your data. Okay. And if you want to do like transcription factors analysis, you can use a transcription factor database. Or if you want to do gene family analysis, you can target the gene families. Okay. And later on, it depends what you want to do with your data. Now, what are the challenges that we face in aligning the reads, the mapping reads to the reference genomes? Because we expect lot of sequencing errors in between also, okay, and polymorphism, that we have polymorphic genes. So sometimes they don't align to the reference genome. Okay. It is more difficult to map reads that span splice junctions. Okay. So if you have a splicing, and then reads will not align, because when we read the books, we say that if you align the DNA with the mRNA, mRNA will bind at exons, and where there are introns, the DNA will make a loop, but the software will not make such kind of loops. It will simply throw out the read from alignment, because this is not matching with this. Okay. Then repetitive sequences, because in our DNA, we have lots and lots of repetitive elements, retro transposons, bhare pade hue hain. Okay, they always create a problem, starting from sequencing to assembly to analysis. Now in sequencing, because everything is dependent upon, except this nanopore, everything is dependent on PCR. Okay, so suppose you have a repeat of like 80, 80, 80, it is coming repeatedly 10 times, 20 times, then the polymerase get confused. Okay, then it may skip some of the 80s or it may add some of the 80s in your data. Okay, so this is one problem. Suppose it has accurately sequenced that. Suppose there are only 10 80s in the data and it has generated only 10 80s and that are correctly sequenced. Then the problem comes in the assembly of the data. Then you, because you are not sure how much is the repeat size, so you simply software says कि आपने 10 overlap करने हैं, आपने 15 overlap करने हैं. So it in the assembly also the repeat may be shortened down or increased. एक्चुअली में वो आपके जीनोम में नहीं होगी या ट्रांसक्रिप्टोम में नहीं होगी. ओके, देन बिकॉज दीज थिंग्स व्हेन यू एनालाइज द डाटा, है ना, जब हम ब्लास्ट करते हैं वी ऑलवेज टेक मास्क द रिपीट्स, रिपीट मास्किंग ऑल्सो वेल देयर, कि जितनी भी रिपीट्स है एलिमिनेट दैट फ्रॉम डाटा एनालिसिस. सो व्हाट इज द मीनिंग ऑफ दैट? बिकॉज व्हेन दे विल कम देन दे विल नॉट मैच विद एनीवन एंड यू विल हैव लो स्कोर ऑफ द अलाइनमेंट. आपकी अलाइनमेंट जो है वो कम आएगी एंड आपकी जो फिर हो सकता है सिग्निफिकेंट मैचिंग को भी नॉन सिग्निफिकेंट कर दे ऑन द बेसिस ऑफ स्कोर एंड वो आपकी अलाइनमेंट से बाहर हो जाए. ओके, सो ऑब्टेनिंग लॉन्गर सीक्वेंस रीड्स और पेड एंड सीक्वेंसिंग स्ट्रेटजी, दैट्स व्हाई वी ऑलवेज सीक्वेंस फ्रॉम बोथ द एंड्स, दैट you should be highly sure that whatever you have generated is of good quality. So if you have generated a good quality sequencing data, everything will be go good. अगर आपने पहले से गलत कर लिया तो आप कुछ भी कर लो, everything will come wrong after that. Okay, so if you want to analyze families, okay, like suppose nowadays many people they try to find out the gene families whom they are interested in. Okay, we also do the same. Okay, if you want to increase the number of publications, you can analyze the gene family in your plant and publish it. Okay, so what do you do in this case? Suppose you want to find out how many genes of particular category they are present in your genome or in your transcriptomes. Then what you take? You take a reference sequence. If we are working on plants, so Arabidopsis, rice, and 3-4 more plants you can take. You can take their sequences and then align with your data. Okay, and जितने भी आपके उस कैटेगरी के जीन होंगे वो सब निकल के आ जाएंगे. Okay, so suppose you have 25 sequences of that, or 100 or 200, sometimes you have 700, 800 sequencing of one gene family. Okay, all these things will be clustered together with the reference genome. आप Arabidopsis लेते हैं, ठीक है, as a standard, wheat लेते हैं, rice लेते हैं, हम लोग you come to know कि आपका closest relative कौन सा है plant का, we try to take that also. ठीक है, फिर उसके साथ अपना ट्रांसक्रिप्टोम अलाइन करते हैं एंड देन जितने भी हमारे सीक्वेंस निकल के आते हैं एंड फिर हम उसके साथ सर्च करके जीन फैमिली आइडेंटिफाई करते हैं. देन यू फर्दर कैरेक्टराइज देम. देन यू ट्राई टू फाइंड आउट इफ ऑल द सीक्वेंसेस दैट यू हैव isolated from the transcriptome data, they have a particular signature of that family. So you do domain analysis of that. Okay, you try to find out the domains. If they are present in that family, then it's okay. If in some sequence that domain is not present, then you simply remove it from analysis because that is not having that signature domain in the data, then it's thrown out. फिर आप सारी बाकी एनालिसिस करते हैं, उसके phylogenetic analysis, their expression. Okay, अगर आपको उसको characterize करना है to the largest extent, then you can make recombinant protein out of that, then you do enzyme assay, or you can make construct of that, put into plant and make the transgenic one, or you can do transient assays. Everything is possible in that case. So I think that's all for today's class. Any question you have, you can ask. First thing is you know better than the software. Okay? Your analysis power is better than the software. So whatever you tell computer, it will do only that thing. So if the two computers they are so two tools they are giving some contrasting results, then you use third one. Okay, and check what third one is saying and उस दोनों में से जिसके साथ मैच करेगा, go with that. ठीक है.